Hello, everyone, and welcome to the August Variety Hour. Thanks so much for joining us today. My name is Talia Speaker. I'm Executive Manager here at Wild Labs, based at WWF in the US, and I will be your host today. We're really excited to be joined by some great speakers and have a good crew joining us. So, we are very excited to be joined by amazing speakers today. We're going to hear about biodiversity around me from Camille, about fin whale migration from Luis, about developing bespoke telemetry for pangolin conservation from Elio, and then our main talk will be from Jennifer on technologies for assisted reproduction in wildlife. And we also have some fun community updates related to this on our new groups and group leaders this year. So with that, I will hand over first to Camille to get us started. Take it away whenever you're ready.
Okay, so hello everyone. So I am Camille Monchicourt. I am a IT manager at the Ecrins National Park in France, and I will talk to you about BAM for Biodiversity Around Me, which is an open source widget that we created to make biodiversity open data easily accessible to everyone, everywhere. So the Ecrins National Park is one of the 11 national parks in France. This is the purple one in South Alps, and we are managed by the Ministry of Ecology, and we also work closely with the Biodiversity French office. And protecting the knowledge of biodiversity is one of our main mission, and that's why we have built and developed several web applications and databases to collect, manage, and share our data about the biodiversity of the national park. But we did it mainly with another national park, the Seven National Park, in an open source model and generic, so that the tools that we do for ourselves could be used by other organizations. And that's how we have built communities, because we really aim to save the world all together. That's a hope. And after several years, we had the observation that most of these data are open. They are usable by experts, scientists, naturalists, with platforms like GBIF or also web atlases where you can get the data. But there is still a lack of simple accessibility to all this data for the general public that is not an expert or knows all this kind of tools or data. And that's how we started thinking and concepting the tools, the tool BAM. We saw about it as a widget that could be very easy to include in some website or web application to make the data very easy to access. The idea is like maybe you know the weather widget that you have on several websites that are very easy to include and that you can see in several websites. To have the same thing with biodiversity, maybe one day the people will be interested by biodiversity as much as weather. I'm not sure, but we hope also. So that's the first development session that we had with the Seven National Park and a few other national parks in 2024, where we're starting developing this tool. So now how it works. This is a more complicated schema, but I will explain. First, as a widget, there is a web page where you can configure the widget. You give all the settings, like the color that you want the widget to be displayed, the way the information will be also displayed with a map or not, the data source if you want to connect to the GB for other source, and also, of course, the location, because it is based on the location of the information that you want. It will give you a simple HTML code as an iframe, as you can see below, and you can just copy paste this iframe code into your website, the same way you include YouTube video in an article or a page or a weather widget, as I said before. Then, when the page will be displayed, the widget will automatically connect to the data source. The main one is the JBIF. It will retrieve all the observation in the location that you have selected. It will also get some media and images to have a good layout of all the species from the JBIF or from Wikidata or other source. And it will display the list of all the species in this location. So you have nothing to install, no server required or no database, because it will connect to API, and it is multilingual in French, English, Spanish, but also thanks to the open source model, we got some contribution, and now it's also available in Czech, German, Italian, and we hope other language soon. Just a few examples. This is one of them. This is an example of the widget integration. This is a web page where you have a map of the schools in a German town. And when you click on one of the school, the BAM widget on the side will automatically get the location of the school and find all the observation in 500 meters around and display the list in a simple way, easily understandable by anyone, of the species observed around the school. Another example in a trek website where you have the treks, the track, and all the information about the trek, but you can also include the BAM widget inside the page of the trek, and you get all the species observed around the trek to give additional information about the biodiversity for the people that will go to do some trekking there. Or this is another example in a simple web article that can be on the media, or there it is on our national park website, and you include it as a YouTube video, but this time with the list of species in a location that you choose. It can be an event, an accommodation, or anything else, and you will have the list of all the species observed around. Finally, BAM can also be used as a standalone web or mobile application. You can use it in a widget included in the website, but also as a global application. You just go on bam.geonature.fr. You will be located, and the same way it will retrieve data from GB for other source, get all the species around you, and list all the species in the area around you. And you can move anywhere in the world the map to search for the species anywhere else. So that's it. To conclude, we try when any time that we create tool for our national park to do it with an overall approach, so let's say with open source and generic approach, to simplify the data access and share our tools, the more generic and the more open as possible, to improve the knowledge acquisition, the sharing, and the simulation of the data. And I thank you for your attention. I hope that I was good for the timing, but let me know
Yes.
if it okay.
You were great. Thank you so much, Camille. That was super interesting. We have time for maybe one or two quick questions. One for me was just, I'm curious to know what kind of feedback or collaboration or input would be most helpful from the Wild Labs community.
First of all, the most important for us is that it is known and used as much as possible. So I really thank the WildLab network to help us to let this tool known for as much as possible. But if you want to contribute, maybe you can add some settings or some languages. As I said, we have 6 languages. Maybe it can be interesting to have other languages. But the most important for us now is that it is used as much as possible in your website, in your web application, or otherwise just on your phone, and you tell it to all the people that know nothing about this kind of data, or also about biodiversity, but has an interest or create an interest. So we look for some promoters and for users as much as possible. Yeah.
Very cool. It looks like we have one question from Allard. Allard, do you want to jump in and ask your question?
Sure. Bonjour, Camille. Je suis aussi
Bonjour.
francophone, mais bon. I will ask my question in English.
All
Thank
right.
Does
you.
it in the US we use a lot, or I at least personally do use a lot the Merlin ID, which is very simple, but just for birds. Um, can this integrate in theory with that? It was developed by Cornell University.
The the default use case is to connect. BAM is by default connected to GBIF data. So as soon as your data are inside the GBIF, they will um, they will be into BAM when you look for a place anywhere in the world. If you want to connect to another data source, this data source must have an API, and then if you have a specific list of species or anything, as soon as you make the connection with your data and your source, it will work. But you have to connect it to your specific data, and that's something to develop. But the most simple is that your data is included into GBIF, and by default, bam, connect to GBIF, and it will work this way.
Thank you so much, Camille. We really appreciate it. Feel free to follow up in the chat if anyone has more questions for Camille. All right, so next, our next speaker is Luis, who will be talking to us about a collaborative approach that challenges fin whale migration assumptions off of South America. So Luis, whenever you're ready, please go ahead.
Well, thanks for having me. I'm Luis Bedriñana from Copas Coastal, Universidad Austral de Chile, and Blue Whale Center in southern Chile. I'm going to start saying that we live in times where we have a lot of toys to play with baleen whales. We have these cool tags that allow us to follow them for several months to inspect their migratory patterns. We have these other fine scale tags that allow us to describe their diving profiles in very detailed manner. And they also have cameras that allow us to see what whales see, how they feed. We have acoustic gizmos that allow us to detect their acoustic signals across several parts of the ocean, even gliders that can detect their songs and simultaneously sampling for oceanographic data. We have vessel tracks, the good old-fashioned vessel tracks that allow us to inspect the distribution patterns visually. So what we did in this work is, what can we learn about fin whale migratory pattern if we put all our toys together, all our data together, and try to learn more? What we knew about fin whales is that they migrate from the Southern Ocean, where they feed in summer, to someplace in subtropical areas. So if we start by seeing what we can learn by putting all the visual surveys together, is that here you can see in green are the navigation track from the vessels, and in yellow you can see fin whale sightings. Immediately you can depict that in the Humboldt Current ecosystem, which is the part of Chilean coast, there are a lot of sightings. They are present during the entire year, and in the coast of Brazil, in Rio de Janeiro, where a lot of vessel effort have been conducted, there's also a concentration of sightings. The difference is that in Rio de Janeiro, they are only present during winter. So just by seeing these two patterns, we can see that at least for the Atlantic part, this agrees with the classic migratory pattern, but in the Humboldt Current ecosystem, this does not agree with the classical migratory view. They are permanently there. They are the most abundant baleen species in the Humboldt Current ecosystem, and they are pretty much everywhere. And in the other areas, like in the Argentinian platform or even in Chilean Patagonia, you can see that sightings are very scarce, except for this area which is the Namuncara Bullwood Bank, which is a marine protected area from Argentina, where there are recent sightings there. Now let's move to what the acoustic data says. Now we have a lot of information here, but let's concentrate on one thing. In the x-axis, you can see the months of the year, and if the bar is pink, it means that the acoustic presence of fin whales is there, and if you see light blue, it means there are no acoustic detections. So here, this agrees with our visual service data, because in the Antofagasta and Chañaral and Juan Fernandez station, which are in the Humboldt Current ecosystem, acoustic detection occur during the entire year. In Rio de Janeiro, they are detected mainly in winter and austral spring, again in accordance with the classical migratory patterns of this whale. And in Puñihuil, which is this blue dot in northern Patagonia, you can see they are only present during winter, which it's gonna make much more sense in the next slides. So again, we have data that supports a classical migratory movement, and data that says that at least a part of the population does not migrate. And this is how we come to this novel part of the telemetry data. We tag our whales in the marine protected area Humboldt Archipelago, which is a really small red dot over here. And here we can see that most of our whales spend most of their time in the Humboldt current ecosystem. And the few whales that have been tagged in Brazil start migrating to some place because the tags stop transmitting during winter, which again agrees with classic migratory patterns. Now, let's focus on this particular guy that we tag in Chañaral de Aceituno in the northern part. You can see in the animation. Every time the whale track turns red, it means it might be eating, and in blue, it's transiting. And these guys spend most of the time in winter and autumn and summer in Humboldt current ecosystem, then move to the Namuncurá bulbul bank. And if you see a flying dot, it's not evidence of alien abduction. It's just that the whale tag stopped transmitting. And this is very interesting for several reasons. First, this whale start migrating in winter from a mid-latitude area to a high-latitude area. And this is the first description of a inverted migration, if you might say, because as you know, whales are supposed to migrate in the other direction. They are supposed to move to low latitude during winter, after winter. And this guy did the exact opposite. And it's also the first description and the round trip migration of fin whale in the entire southern hemisphere. So this was very interesting. And the other thing that is important is that this guy moved from one marine protected area to another marine protected area. And this gives value to marine protected areas. So to sum up, we have evidence of the existence of multiple migratory strategies for fin whales, which is in accordance to what had been recorded in other populations in Northern Hemisphere. We have the first description of an inverted migration, which is novel for all whales, as far as we know. And the importance of migrating from one MPA to another one is very important because this is why we create marine protected areas, not just for several ecosystem processes and species, is because they have secrets that we don't know when we create them. And this is very important. And finally, I think in times where science is pretty much under threat and budgets are being cut and people are like skeptic about climate change and stuff like that, collaboration is key. Even when this is always important, collaborating is now perhaps more important than ever, especially in developing countries. A work like the one that I have presented to you right now that covers vast parts of the ocean in an entire continent is only possible if we collaborate to each other. So that being said, thank you very much for your attention.
Thank you so much, Lisa. Super interesting. Really fascinating case study of working out across borders, across geopolitical borders with multiple methods. I think there's lots to learn there from that work. Unfortunately, we don't have time for questions now, but please feel free to ping any questions you have into the chat, and hopefully Luis can follow up with you there. Thank you so much. All right, our final speed talk is from Elio, who will be talking to us about a bespoke telemetry solution for pangolin conservation. So Elio, take it away when you're ready.
Hi, everyone. My name is Elio Challita. I'm a roboticist at the Harvard Microbiology Laboratory. And this project is led by myself and Charles Imogor. He's a conservation biologist at the University of Cambridge. And our goal is to study white and black-bellied pangolins in Nigeria. These are among the most heavily and hunted and trafficked pangolins in the world, and we still don't know much about them. And a major reason behind that is that they are very difficult to track. They live under dense canopy. They are nocturnal, they climb and burrow, and any device we attach has to be lightweight while lasting for weeks. So existing methods, they have limitations. GPS doesn't really work that much under dense canopy, and VHF is only accurate for like around 100 meters. And there's a nice paper that was written by Charles that outlines this work. Sorry, I'm in the back. If you're interested in understanding more about challenges we get from tracking these animals in the forest. So our goal is to achieve long-term accurate tracking under dense canopy with limited mass and power budgets. So instead of looking at off-the-shelf systems which do not really meet these requirements, we are building our own custom tag that is designed specifically around pangolin anatomy and behavior. It is very hard to access pangolins. So what we did is that we went to the Harvard Comparative Zoology Museum and we built our own kind of replica of how the pangolin scales look like. And this is kind of a replica that shows a pangolin skin. And we built our own tag, which is around 50 grams and combines GPS and IMU. It is mainly, the tag is mainly turned off when the pangolin is not moving, and it turns on when it's doing something useful. And it takes a GPS and constructs basically the movement based on both the IMU and the GPS. And we're using dead reckoning for that as well. Now for data retrieval, what we are using is basically we are attaching VHF and LoRa to a drone where we set a base camp in the forest, and over long distances we use VHF, which is proven to work in the forest, but it will give us a rough estimate where the animal is over longer ish distances. And then we take our drone when we get closer to the pangolin. We establish LoRa communication, which is basically like a low cost and low power, long range wireless communication. And we can upload the data we have without really recapturing the animal. And we can process all the data off board without really computing anything that is heavily done on the tag itself. And I guess one of the key advantages here is that we don't have to recapture the animal. And we tested this in the field recently. If anyone is from the Vancouver area, I saw someone is from the Vancouver area, they might recognize this hike here. This is in Joffrey's Lake in BC, and we tested it with different kind of intervals of GPS fixes. And we saw that, if we have a fix every 1 minute, we can get a very good estimate of where the tag is, and then the error would increase to the power 2, actually, to the time to the power 2, if we increase the fix to like 10 minutes or 30 minutes. So in a way, we can estimate the amount of error we can get from this, and depending on what kind of information you want to get from the tag, we can change the duration of our tags. And we're gonna, this is still under development, and but I think we're pretty close to deployment. But yeah, if anyone has any suggestions or any questions, I'm happy to answer these. And yeah, thank you.
Wonderful. Thank you so much, Elio. Super interesting. So one of the questions that I had for you is just, it's such an interesting process having like the 3D printing, like being able to actually look at the scales to work with the, but to see like, how your technology is going to work developing that. And I'm curious if that's like a common practice in developing attachments for hard to track species, or if there are maybe things from your development process that could be transferable to other species that are difficult to attach trackers to.
That's a great question, and I think that it is not typically used for building attachments. And for us, when we start working on these, like we want to really work. I haven't seen a pangolin yet, and it's hard to find them in zoos here in the US. There are only a few spots, not in the area at least. And so we were like, all right, how can we build something that is informed by the anatomy? And then we started looking at what are the available resources. We have videos and we have samples from the Harvard Museum. And then we thought like, okay, this is actually interesting. Maybe we can use that to make something that is more generalizable beyond the pangolin. So we are actually writing a paper and a method paper on how to use video information and museum samples to reconstruct basically the animals without having them, basically based on their gait and how they move in the forest and so on. It's a little bit in parallel to what our main goals are, but I think it would be interesting to the community as well.
Interesting. Yeah, please do share that when it's available for the community to read. Wonderful. Well, thank you so much, Elio. All right, so that wraps up our short talks, and it is now time to jump in to a couple of quick community announcements, and then our quiz. So first, we're super excited to welcome our 2026-2027 Wild Labs Group Leader cohort, which we just announced. This is the third cohort of the Group Leader program, and this year we have 31 members of the community leading 12 different groups here on the screen, covering everything from acoustics to animal movement, AI for conservation, eDNA. You can see the rest here. Including, yeah, topical and also regional communities. So a big thank you to everyone who is volunteering their time there. We think that this is a really important program that helps us kind of strengthen our community and also just let us do more than our small team can do directly. So please do go check out those groups and engage. A lot of the group leader activities are focused on hosting events, so keep an eye out for group leader-led events as well coming up. And then along with that, we are really excited to share that we have a new group that's launched on Wild Labs focused on zoos and aquariums. So this will include topics like in situ and ex situ work, including like animal welfare, behavioral research, tech transfer. There are a lot of really interesting case studies like shown in the picture here, where zoos can be used as really helpful testing sites before tools are deployed out in the wild. So yeah, we're really excited to bring this conversation into the community. I think it's been captured in some ways in other groups, but we are excited to have a dedicated space for it and really wonderful group leaders to take the lead. And then it's also a really nice connection to our long talk today from Jen, who's going to introduce us to reproductive technologies, which are another interesting area that group will focus on. So yeah, go check out the group, join the conversation, and yeah, feel free to contact the group leaders if you have any questions about it. Cool. Now it is time for the quiz, which I have to say, Vadne, our team member Vanessa Reyes, did this quiz, and I'm taking over for her today. So all credit to her for the content of it. The focus today is going to be on El Niño, which is a very topical thing in our world right now. So the first question, so please feel free to just put your, like always, put your answers in the chat. And then can someone from my team help me look at the chat?
Yes, we can.
Okay, thanks, Adrian. All right, so the first question is, where does the name El Niño come from? A Spanish term introduced by meteorologists in the 1950s. B, Peruvian fishers who noticed unusually warm coastal waters around Christmas. C, a traditional name for storms in Ecuador. D, the name of the first research vessel to document the phenomenon.
You have a lot of B,
Okay.
some
Oh wait,
D,
actually I
some
can see
A.
this.
Oh, you
Thank
can
you.
see it?
I actually can see it now. For some reason it wasn't showing up before. Cool. Perfect. Yeah, lots of B's. All right, I think we're on to something here. The answer is, in fact, B. Peruvian fishers used El Niño, referring to the Christ child, for unusually warm coastal waters that appeared around Christmas. The term was later adopted for the much larger Pacific climate phenomenon. Nicely done. All right, next question. Which of these technologies can scientists use to monitor El Niño? A, remote sensing, B, moored ocean buoys, C, autonomous profiling, floats, or D, all of the above. D, 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 D. I would suspect that that's the correct answer, and in fact it is. Scientists combine remote sensing and in situ observations to monitor El Niño. Remote sensing provides broad surface coverage, while buoys and profiling floats measure conditions at and below the ocean surface. Fishing one, two. Cool. All right, next question. In July of 2026, how much warmer than average did NOAA observe some waters below the surface of the equatorial Pacific? Up to 2 degrees Celsius, up to 5, up to 10, or up to 15 degrees. You got a range here, B, C, A, A's, B's. Seems like more B's than anything else. All right, let's see. The answer is C. So I guess no observed temperatures reaching 10 degrees Celsius above average below the surface in parts of the equatorial Pacific. Super interesting. And these are monitored using an array of moored buoys. All right, next question. During the 2014 to 2016 Pacific Marine heat wave, why did humpback whale entanglements and fishing gear reach record heights off of California? A, warm conditions compressed productive upwelling habitat towards the coast, increasing overlap between foraging whales and crab fishing gear. B, warmer water interfered with whale tracking devices, making it harder for fisheries to avoid them. C, prey became so scarce that whales began feeding on bait and commercial crab traps. Or D, warmer water caused whales to remain on their breeding grounds longer, overlapping with crab fishery. Let's see. We have a lot of A, some D, some C's. Seems like majority A. All right, let's see. The answer is A. Nicely done. Researchers found that the marine heat wave compressed cool upwelling habitat towards the coast and altered prey availability. Interesting. All right, next question. During the 1997-1998 El Nino, how much did new primate production decline in Central California waters? 20%, 40%, 70%, or 95%? D question mark, C, B, C. Seems like we're split between C's and B's mostly. All right. C. A study found that the surface nitrate fell to less than 20% of normal, while new production declined by close to 70%. High nutrient productive waters that normally extend from the coast to about 200 kilometers offshore largely disappeared during El Nino. All right, I think this is our last question. How are researchers reconstructing the effects of past El Nino events on wildlife at Palmyra Atoll? Historical satellite imagery, sediment cores containing pollen shells, isotopes, and biomarkers, archived camera traps, or GPS tags on living seabirds? Lots of B, sediment cores. B, B, B. All right, I think we got it. Yep, B. Researchers are using sediment cores and biological and chemical proxies to reconstruct past climate, vegetation, and bird populations, allowing them to investigate ecological responses to El Nino over timescales far beyond modern monitoring records. Good job, team. Thank you so much for participating, and thanks to Vane for putting the quiz together. All right, let me just close out of this. Okay, so now we are on to our last talk of the day, which is our longer talk from Jennifer. And as I mentioned, we're excited to have Jen's talk today because it's linked with the new zoos and aquariums groups. So Jen's going to share with us about technologies for assisted reproduction in wildlife. Jennifer, whenever you're ready.
All right. Well, thank you so much for the invitation to meet up with you all today. I'm really excited to chat about assisted reproductive technologies for wildlife conservation. So the first thing I want to bring up is what are assisted reproductive technologies? I'm actually coming to you from the zoo world, the Smithsonian's National Zoo and Conservation Biology Institute and the Center for Species Survival. And this is really our bread and butter is understanding reproduction in wildlife and developing these techniques for species survival. But I know it's not as familiar to everyone. Basically, these are any procedure or treatment that is used to achieve pregnancy and healthy offspring, really core to our ability to conserve species. One of the examples from the human world that you might be more familiar with is in vitro fertilization, like human IVF clinics. So this is where infertility diagnostics might take place. And then if a woman would like to go through an IVF cycle to donate her eggs or to produce those eggs, she needs to take injections of hormones to get her ovaries to respond and grow additional eggs. And those are collected, put in a petri dish with sperm to produce embryos. So this is some images of actually dog IVF, not human, but as an example. And this is pretty well established in the human space, but as you can imagine, there's a lot of challenges that require innovative and outside of the box thinking if we want to translate even this quote unquote basic technology that we use every day for our various wildlife species. So one of the key things that is maybe different is that we don't have a lot of information about the basic reproduction of a lot of our species. So on the right-hand side here is a very detailed graph of the human menstrual cycle, reproductive cycle over the course of 28 days, where we have data about the actual status of the uterus, the endometrial glands, and then also the really dynamic changes in hormones that are circulating through the body that's helping to grow these eggs and then maintain the pregnancy, as well as data from probably ultrasound work showing how the ovary is growing and that egg is eventually ovulating and then producing a structure that creates the progesterone to maintain the uterus and maintain the pregnancy. So this is all from, blood serum data from humans and ultrasound data on a daily scale that we really don't have to the same degree for most of our species. So this same figure might actually apply to some of them, like non-human primates, but this is not the same whole internal biology that is being experienced by most other species. If you want to translate this to a giraffe or a pangolin, we need to have these data before we can develop something like those injection protocols to get eggs, because otherwise you could actually be hurting the animal if you do something inappropriately at the wrong time or the wrong dosage, right? So we need this information. So I actually really appreciate that Louise and Elio were talking about all the cool new toys that are available for monitoring the movements and behaviors of animals. What I want to introduce here is some concepts, including how we might be able to monitor internal biology, because, seeing how they move is incredibly important, such a valuable tool. If we can understand also what's going on inside the animal at the same time, maybe we can better understand why they're moving in certain ways by monitoring that as well. So the way I've set up this talk is introducing 3 big themes in a very short amount of time, and then some current and what I hope are future technologies that I'm really excited about for their application for wildlife conservation. So one of the current technologies that we have is called this the gold standard, and you might have heard of it. We call non-invasive hormone monitoring. So this is some work that I'm highlighting from one of my colleagues at NZCBI, Dr. Janine Brown. And that ovary I showed you from that first picture that ovulated and was producing the hormone progesterone to maintain the pregnancy, those hormones are actually also getting into the bloodstream, and then metabolites of them are ending up excreted in this animal. So in fecal samples, in urine samples, even hair and saliva, we can actually extract these hormones and get a sense of what was circulating in the animal internally on that day. So this is an example of a graph on the bottom left-hand corner here of a black rhino who bred back at the -15 on the x-axis here. That was a mating time. And she became pregnant, which we can tell through this increase in fecal progesterone levels that are maintained across gestation. And then importantly, too, for zoo management type applications, we can also tell when she gave birth because there was a drop in that progesterone right before parturition. And this is really valuable because if you were monitoring this the whole time as in a zoo or some sort of wildlife management area, you would have a couple weeks notice of when she's about to give birth. And in terms of being able to prepare for that on the management side, it could be incredibly valuable. So that is one tech. It's not monitoring in real time. It's after the fact, and we still have to collect the poop. But something to think about with the group that's doing bespoke technologies. If we can do this somehow internally in real time in the future would be fabulous. In the meantime, some of the other layers that we are adding on to this include really more expansion of what the profiles are like in the internal biology of these animals. So this is some work by Dr. Molly Corder from C2S2 and part of the larger Rhino Resilience Project. And from a same fecal sample from a black rhino, she can extract DNA and do plant DNA barcoding and figure out what diet that animal had and compare in situ versus ex situ housed animals' diets. Also, how those diets might be impacting gut microbiomes, and subsequently through blood samples, looking at the chemistry of the blood, the metabolome, and one really cool project that she worked on, and I should have posted the paper on here, was looking at the proteins of immune cells in black rhinos in zoos versus in the wild, and trying to see if she can identify any pathways that might be dysregulated. And basically by combining all these information together and also integrating some machine learning models, you can start to really get a sense of the layered changes that are happening internally, and how genetics and epigenetics of the species are informing their responses to diet changes or changes in the environment, and potentially either predict how they're going to respond, whether that's in their health or in their reproductive output, because those are two very closely interrelated aspects. Or even develop new treatments if you do have an animal that is not feeling well. So I'm really excited about how these different sets of data are being integrated and are basically creating a digital twin or an omics twin of an animal that we can use for future research. Another thing that I've listed under this internal biology aspect that is a current and future tech is more on the human side right now. But if you've heard of this, it's interesting and also a little terrifying. The idea of implantable microchips that can deliver birth control. So these are programmable type systems, other drugs as well, but this one's gained a lot of attention in the news, that if a woman decides she wants to halt her birth control, she goes on the app and shuts down her contraceptive implant and is ready to go. So there are a lot of ethical concerns with this, biohacking concerns, obviously, but I'm really interested in seeing where this goes on the human side, because it could be very useful for both in situ and ex situ housed animals to either monitor biology or control reproduction in a more fine-tuned scale. So rather than having to capture an animal, anesthetize it, remove an implant, add an implant, if you already had that implant in and it lasts for many years, and you can just program it on your cell phone or through remote drones, somehow deliver that message, that would be a really, really fantastic tool to be able to manage species. The second big concept that I want to bring up is cell cryopreservation. So this is the preservation of living cells at sub-zero temperatures, typically in liquid nitrogen, that brings them down, basically slows their metabolism down to the point that they're in stasis, and then they can stay that way for decades, forever. This is some sample of sperm that I collected from a red wolf a couple years ago. We had it cryopreserved in liquid nitrogen for a few months, and then I thawed it, and this is 4 hours after thawing. And as you can see, these cells are very much still alive, ready to fertilize, ready to go. And so this is something that we can use to help manage our genetic populations. It's a backup bank of genetics for species if we have these living cells available in case of a disaster, in case they need to be reinfused into the population later. At the Smithsonian, we have a Dorrance Hamilton cryorepository. Each of these tanks here in silver contain liquid nitrogen, and we have hundreds of samples from a variety of different species, from mammals and birds, coral, and a growing collection of amphibian cells. A lot of our samples are sperm, but we do have eggs, embryos, and in some cases, ovarian and testicular tissues, because we actually get a lot of tissues sent to us after an animal has passed away in a zoo. And we will try to collect and rescue as much of that cell population as possible. And if they're not happening to be, already pubertal or in breeding season, they might not have mature sperm or eggs to collect. And so therefore, we are collecting the tissues. One of the current technologies that you might have heard of that is a really exciting application of this cryopreserved cell bank is the production of cloned black-footed ferrets. So these were from some fibroblast cells that were cloned by the San Diego Zoo, the frozen zoo, from a founder individual who did not successfully breed. So she did not contribute her genetics to the current zoo managed population, which is now a huge concern, right? Because you want to have the maximum amount of genetics, right? So they wanted to bring her genetics back into the breeding population, back into the reintroduction gene pool for the future. And what the scientists from Biogen and Revive and Restore and this larger collaboration did was take the nucleus of one of those somatic cells after they thawed it, after they took one of those fibroblasts, removed the nucleus of a domestic ferret egg and reinserted the nucleus of the black-footed ferret into that egg, activated it, produced embryos that were transplanted into a surrogate, and produced now 3 of these cloned black-footed ferrets. And importantly, one of these females actually successfully bred naturally, and now her offspring and her genetics are represented in the population. So really fantastic. You've actually infused new genetics from a critically endangered species for which there were no additional genetics available, but really depended on the fact that you had frozen cells and you had cells frozen in a way that they survived the process and were able to develop, which does take some fine tuning and often innovation. And I will take a second here to say that there's a lot of opportunities to advance this. Particularly using AI to optimize protocols. And so if folks are interested, that would be a really great area of new collaborations. Last thing on this cell cryopreservation thing is this idea from one of my other colleagues, Dr. Mary Hagerdorn, who in collaboration with a lot of other groups that I'm not really focusing enough on right now, but basically the idea is to create a backup biorepository on the moon. The idea behind this is that we have our, banks in the facility here that require a lot of liquid nitrogen, a lot of infrastructure, bells and whistles alarm to make sure they're secure. But there is a crater on the dark side of the moon that is the exact temperature of liquid nitrogen. And this would represent a passive biorepository. If you can get it up there safely and protect it from radiation, then it doesn't need to be maintained. And it is your real emergency fund genetic reservoir for the population of species on Earth. So next year, there's a plan to send some of these first samples in a structured box that protects it from radiation trial run to the International Space Station. So stay tuned on how those results go from these cells. So the third and final little bit that I want to bring up is this idea of in vitro reproduction. So I had mentioned that sometimes we have animals where we get ovarian tissue, but we don't have mature eggs. So what can we do to make eggs from that we can then use to reproduce the animal? And conventionally, you would just stick your piece of tissue in a petri dish and cover it with cell culture media and hope it grows. And that's not what happens in your body, right? This is a schematic on the left-hand side of an ovary where you have blood perfusion that's constantly supplying your tissues with nutrients. You have a lot of different phases that these eggs go through to develop where they're growing incredibly and changing their microenvironment around them to be able to facilitate this growth. And that's necessary to get them from the really, really early stages of eggs, this primordial over on the left side, all the way up to ovulation. You need that whole process of growth and development before you're going to get an egg that is developmentally competent, that's going to produce a healthy offspring. And in parallel to that, that's really kind of overseeing this whole thing is that change in hormones, similar to what I showed you on the human menstrual cycle. This is an example of a domestic dog, all the different hormones. You don't need to know what all of them are, but as you can appreciate here in the different colors, this is a very dynamic process that's happening over the course of many months. And so if we're trying to recreate all of our conditions from the body in the incubator, we also need to recreate this because this is what's managing the signaling in the ovary and helping that egg grow to the healthy way. So this is some work that I'm doing also with some of my colleagues at Colorado State University and my postdoctoral fellow, Dr. Catherine Story. And what we're trying to do is use microfluidics. We've created what we're calling an ovary on a chip that's recreating some aspects of this to biomimetically recreate our ovary in vitro. What we've done is 3D printed a mold, which we can then use to create these silicone-based microfluidic devices that contain multiple channels. These are connected to these needles here in the bottom of the top right-hand figure, and seeding our ovarian cells in the center chamber. And then we can flow our culture media, similar to blood flow, over our cells and tissues. And each of those different channels are connected to a different syringe on a syringe pump, pictured on the bottom image, where we can program that to actually do the pulsatile exposure of hormones of a natural reproductive cycle in our microfluidic chip. And the really cool thing about this is that, if we have that baseline internal reproductive biology information from, say, the endocrinology non-invasive hormone monitoring, we actually have that fine-tuned understanding of what's going on in the animal that we can then recreate in here. And with a 3D printer, we can 3D print different ovarian architectures or different tissues as need be within a couple hours and recreate as closely as possible some of these different aspects of the internal biology in our incubator for downstream applications. The idea is that we'd eventually be able to produce healthy eggs that we can use for in vitro fertilization from these systems, even from immature tissue. So with that, I'm going to thank everyone, and if there's time for any questions.
Wow, my mind is blown. I think a lot of our minds are probably blown. Um, we do have a few minutes for questions. I wanted to just kick us off with I think you did a great job of pulling out some really amazing examples of the kinds of things we can do with this technology and even opportunities where, folks would get engaged in one challenge or another. But I'm just curious, now having, I think Catherine's even one of our group leaders, now having
Yes.
this new space in Wild Labs, where do you see the biggest opportunity for this broader kind of conservation technology community to engage with this space?
Oh gosh. I really struggled with this, and as you can tell, since I went over time, there were so many different areas that I can see value in. I think, from my understanding of a lot of the expertise in this group in terms of remote sensing and biologging and trackers, I think that's a really interesting area that might um, be able to move more quickly. The idea of being able to monitor the internal biology of the animal, I think that's really fascinating. There's been some work done in like heart rate monitors that already give a lot of information about how the animal's responding to stress or movement or just daily patterns of life. That's really exciting. But if we can also add these different aspects of, reproduction and other health markers would be really cool. But yeah, there's
Yeah.
so many I don't really know how to. Yeah.
Absolutely. No, but it's so helpful to even just have this lens of the work because even we recently ran a movement ecology horizon scan, and that came up a lot, like internal monitoring of internal states and having a better understanding of behavior based on, and even health in terms of disease. But reproduction
Yeah.
never even came up as one of the
Yeah.
applications that should be considered in development of those tools.
Yeah.
So really interesting. Cool. Pessa, I see that you have a question in the chat. Pessa is asking, could the reproductive control microchips unintentionally have effects on natural animal behavior, particularly mating behavior or social interactions?
Yes, absolutely. So this is actually one of the things that I think the Zoo and Aquariums group is really, you kind of alluded to it, Talia. I believe that some of the technologies that we're developing, you know, provided we can, demonstrate safety, um, zoo animals are a great resource for kind of evaluating this. So, for example, we've had some studies here at NZCBI with new GPS collars on different species where we have put them on the animals here and done behavioral monitoring and also collected fecal samples and evaluated some of these, stress hormones or reproductive hormones to see how even just the external backpack or something of a GPS collar is affecting their behavior. So this is definitely something that would need to be tested before it was deployed largely or in the wild.
Okay, thank you. One other question. I don't see any others from the chat, but feel free to pop them in there, if anyone else has them. But one other question I had is just because this new, like Phoenix species project funding from
Yes.
Bezos and then was just announced. I was just curious if this is like seeing the black-footed ferret, example, if that's kind of part of the lens of what that project is hoping to leverage.
I certainly hope so. I'm still
Okay.
kind of keeping an eye on what specific projects are coming out from that. Last I had seen, I didn't really see all the details, but yeah,
Okay.
I'm keeping an eye
Cool.
on it.
Yeah. Seems like lots of opportunity there. Wonderful. Okay. Well, thank you so much, Jennifer. We really appreciate your time, and that's such a wonderful introduction. I think this is a new space probably for a lot of folks in our community, so it's wonderful to have you here, and hopefully there are many more opportunities to connect.
Yeah, thank you for your time.
All right, everyone. With that, I think we can call it a day on today's Variety Hour. Thank you so much all for joining. Thank you to our wonderful speakers. It's great to see everyone. And as always, we'll hang around for a few minutes for after hours for anyone who's available. And otherwise, we will see you next month. Thank you so much.